Lecture 15.
So we use that and then begin lecture 14 that talks about the cells in the nervous system and how they're organized. And the quicker questions at the end today. Okay, the reviews for the Unit 2 exam, we held one before class, and there will be another one after class today for the first part of the alphabet in the large room behind us, LA 124, that we have our split exams in. Okay, this is the concept that really is key to understanding the nervous system that it takes a while to understand. That is that a given neuron receives inputs from numerous sources, as is shown very elaborately on this figure. Neurons have a receptive surface that includes not only the cell body but also all those dendrites. And so they can have thousands, literally thousands of synapses on that surface, all from different neurons. So all these little green worms are not some sort of attack, but it's neurons investing their little opinion on what that given soma should do. And what that neuron there is going to do is summate all the inputs, and some of them will be EPSPs and some of them will be IPSPs, as we said last time. So, of course, the EPSPs would try and depolarize a neuron, and the IPSPs hyperpolarize. And what this neuron must do is sum all of those inputs. So the excitatory endings would end up depolarizing, as represented here, but the inhibitory would end up hyperpolarizing. So what happens finally is a result of the sum of all those inputs. And that is summed right here at this area called the axon hillock. So if that area right there becomes that cone-shaped area that goes into the axon, if it reaches threshold, then that particular neuron will fire an action potential and send information down the axon. So we talk about integration at neurons. This is largely what is being done. There's all kinds of information coming into this particular cell, and it summates that and decides whether to respond enough. Also, there's the same type of integration that occurs in muscles that we talked about before, both spatial and temporal summation. So if you have two EPSPs that happen from two different neurons, input 1 and input 2, individually they could not reach threshold, but when summed, they sum and could potentially reach threshold. That's spatial summation. In other words, having more than one input that has a stronger effect. That's the same thing as a muscle contraction when you start adding more and more cells to have a large muscle response. So we call that spatial summation. It's the same kind of reasoning that we call spatial summation in muscle contraction. And again, the significant area of whether or not this particular neuron fires is this axon hillock region. In temporal summation, instead of having several different neurons fire simultaneously and causing that nerve to reach threshold, if you have one fire repeatedly at high frequency and produce many EPSPs, that should be, and excitatory postsynaptic potentials, not end plate potentials, EPSPs, then you will get potentially a high enough EPSP to reach threshold. And the same thing. So in either case, you can either spatially or temporally summate the presynaptic inputs. This is a summation of what we just talked about. Here is a given cell, and whether or not it fires right here at the axon hillock is a function of either temporal or spatial summation often. And so this is a single presynaptic ending firing that does not reach threshold, so this particular neuron does not send an action potential. If you have several firing simultaneously, you might get it to rise above 30, or again, temporal summation, if you have a few or one of these endings firing up here quickly, you can slowly rise that particular cell up above threshold to get some action potentials. There's also another type of integration called presynaptic innovation, and there's also presynaptic facilitation, and that is when some other neuron affects the presynaptic ending on this neuron right here. It could hyperpolarize or depolarize that neuron and alter how much neurotransmitter is affected on the postsynaptic neuron. So the EPSP amplitude of this presynaptic neuron can be either increased or facilitated based on this particular axonic, they call it, synapse. So you can depolarize that particular ending enough that hardly any neurotransmitter comes out, or hyperpolarize it. So I said that backwards. If you depolarize it, you get more neurotransmitter released. If you hyperpolarize it, you get less. And this is usually due to the amount of presynaptic calcium that gets into this cell and causes neurotransmitter release. So if you, for example, facilitate this presynaptic ending, that will increase calcium inside the ending and increase neurotransmitter release. So this is part of the integration story by presynaptically controlling how much neurotransmitters release. We're going to get into, when we get way into higher functions of the nervous system, learning and habituation and facilitation and all these things on a whole brain level. But we're beginning to understand at a simple level of synapses such things as facilitation and potentiation. And facilitation is following a rapid train of action potentials. You'll get more and more neurotransmitter release. So this is the presynaptic activity on a given neuron. And if you have lots and lots of presynaptic activity, the postsynaptic EPSP's amplitude gets higher and higher and higher. And that's called facilitation, and it occurs for milliseconds, just a very, very brief period of time. So this would be not facilitation, but the amplitude of the EPSP gets higher and higher. That's what we call facilitation. So we're getting more change in that postsynaptic cell by giving extremely high levels of action potential. Potentiation is something that lasts for a period of time afterwards. So the action potentials after a train will have higher EPSPs than they would otherwise. Both potentiation and facilitation are phenomena that are probably due to calcium. In other words, it's a result of conditioning of that presynaptic ending. If you keep stimulating, you're going to have more calcium within it, and so it's going to release more neurotransmitter. And then, even after you stop stimulating it for periods of minutes, often subsequent stimuli will evoke a higher than normal EPSP, excitatory postsynaptic potential. And we think it's due to elevated calcium, although there's some other possibilities, such as the postsynaptic signaling process, intracellular signaling cascades. And the book talks about that a lot. But if you read the fine print, the only thing proven at this time to any level is that these trains of presynaptic stimulation cause that presynaptic ending to have elevated calcium that's slowly taken back up, and it releases more neurotransmitter than would occur without this conditioning. And then even for a long period of time, subsequent action potentials will have a higher than normal level, and that's called potentiation. So this facilitation is during a given strain, and it'll last a couple of milliseconds. Potentiation can last for minutes after. And this sounds exactly the opposite, and it's because if you keep stimulating cells for often this is days to hours to days, you lose the ability of that synapse to have much power, if you will. It's called habituation. So the opposite occurs with long periods of time. And there's all kinds of theories about why this occurs. It's almost like drugs being less effective when given for days and days. It looks like synapses become less effective if you keep using it over and over and over and over again. When we measured, it looks like we ceased releasing as much neurotransmitter than we did previously. The ending is certainly not running out of neurotransmitter. We synthesize neurotransmitter way faster than we can ever use it. But for some reason, it becomes less and less able to release the same quanta of neurotransmitter. It's almost like any type of signaling process in our body. When we keep using it over and over again, it becomes less sensitive. In drug conversations, we call it tachyphylaxis. When you keep giving a drug over and over again, you have less and less response. This is almost like that in the nervous system. The applications of facilitation and habituation and so forth on a larger scale we'll talk about later when we talk about memory and other things that happen in the nervous system. Okay, now shifting on this introductory lecture to different families of neurotransmitters. I have mentioned already that we have many, many different neurotransmitters that we've proven that are present in the nervous system. And this is just an introduction to some of these chemicals. Before we decide something's a neurotransmitter, there are several steps we must prove that that case is true. The presynaptic neuron has to, of course, be able to synthesize and store this substance, so we can now label substances molecularly and find out if that's the case. They must have the ability to release this chemical in response to some stimulation. What's done very often is the exogenous administration, taking that particular chemical and giving it to either an in vitro system or even an in situ system and see if the response occurs. And so you see if you can apply that substance and duplicate the response. And finally, the most used of all these, we have very specific antagonists now for almost every neurotransmitter, so we can find out in which phenomena a given neurotransmitter is responsive, or what if you block that given neurotransmitter, what's the result. So we have some very specific antagonists for different neurotransmitters. There's dozens of different chemicals that have been proven to be neurotransmitters. There's tremendous variety, and as I said before, not only do we have a large variety of neurotransmitters, but many of these neurotransmitters, not all, but many of these neurotransmitters have different receptors that initiate different responses. And given cells have different signaling cascades for the same receptor. So often the response that a given neurotransmitter produces is extremely complex and different from cell to cell. Most of these are very similar to chemicals that you're very familiar with. There's amino acids that act as neurotransmitters, small peptides, monoamines, catecholamine, for example. So we have very common chemicals being released across these synapses that have profound effects. The one which we've already mentioned and that we've studied probably more biologically than any other, once again because it's easy to get to the neuromuscular junction, is acetylcholine, present and released from motor neurons that affects the muscle contraction. This is the structure of acetylcholine. It's extremely well studied. It's released from those motor neurons which we've talked about, but it's also the neurotransmitter of preganglionic neurons. We haven't yet got into the autonomic nervous system and defined what we mean by preganglionic and postganglionic neurons. We'll have, that's the last lecture of the semester, but I'll be talking off and on about the autonomic nervous system. But both the sympathetic and parasympathetic branches of the autonomic nervous system have two neurons, we call it, a pathway of two neurons. One's before a ganglion, an external or peripheral cluster of cell bodies, which we call a ganglion, and the other's after that ganglion, so we call them pre- and post-ganglionic neurons. Well, all of the pre-ganglionic autonomic neurons release acetylcholine. And finally, the post-ganglionic neurons that are parasympathetic also release acetylcholine. So it's a prolific neurotransmitter, and it plays a big role in health, and we talk about it a lot. We've already mentioned that the enzyme that degrades and limits this response is acetylcholinesterase, and it hydrolyzes the two parts of this neurotransmitter into acetylcholine. And as we said before, anything that antagonizes this enzyme potentiates the response. And I mentioned visostigmine and neurostigmine. These are two oft-used drugs that antagonize acetylcholinesterase, and they potentiate, increase the response to acetylcholine in a system. The receptor for acetylcholine is well shown. It's we call it heteropentameric integral protein. What that means, it has 5 pieces that go through the membrane. They're all alpha helices. I don't know why they show 4 here, but it has 5. There's 2 alphas, a beta, a delta, and a gamma. So the reason they call it heto or pentameric is it has 3 different regions, 4 different regions, but 2 of them are alpha. The alpha subunit binds to acetylcholine, and 2 acetylcholines must bind to that to have full effect. So the acetylcholine binding pocket is on the extracellular face of the integral protein, and when it's bound, of course, it non-preferentially allows passage of increased conduction of both sodium and potassium, as we said before. So it's non-selective for cations, and the principal cations, sodium and potassium, are what's principally change conductance. Okay, the second family of neurotransmitters in no particular order are called biogenic amines. The term biogenic means they're amines that are synthesized in biological organisms, if you will. And they have very simple structures, but norepinephrine, you're very familiar with epinephrine, and dopamine are classic examples of these agents. Norepinephrine is released from postganglionic sympathetic areas. So you probably know the sympathetic systems are right system. It's how we respond to emergency activities. And you probably already know that norepinephrine is a stimulatory type of agent. It increases our excitability and our responses to most things. We'll talk about it in detail later. We also, another catecholamine in the same family is epinephrine, and it's released only from the adrenal medulla. So, and the adrenal medulla also releases norepinephrine. So these catecholamines, we call them epinephrine and norepinephrine, are released from the adrenal medulla as well as postganglionic sympathetics. Serotonin and dopamine are CNS neurotransmitters, central nervous system neurotransmitters, released from neurons throughout the brain, and also have very similar characteristics that are increased in excitability. We have some very classic amino acids that also serve as neurotransmitters. Glutamic acid is actually the most prevalent excitatory neurotransmitter in the brain. We talk about acetylcholine so much that you get the idea that that's the primary neurotransmitter. Certainly not. Within the brain, glutamic acid is far more important than any neurotransmitter. And then there's a couple of inhibitory transmitters, GABA and glycine, that both increase chloride conductance, make chloride the most permeant ion in neural tissue. And remember, chloride has a Nernst potential very close to potassium, so it hyperpolarizes and makes cells less excitable. So GABA, which stands for gamma-aminobutyric acid, right up here, and glycine are very important inhibitory neurotransmitters. Might have heard about barbiturates and benzodiazepines that are used often in people that are depressed and despondent, these agents bind to GABA receptor site, and they're agonists. The word agonist and antagonist is often confused, but agonist is an agent that couples to a given receptor and has the same response. Sometimes antagonists bind to the receptor but block anything else from binding to it. Well, benzodiazepines and barbiturates just do the same thing that the native neurotransmitter does. They have the same response. And so they enhance chloride conduction, which means that they kind of make us mellow and relaxed and less depressed often. We'll talk about some other agents in these same families that play a role, modulate neural activity, we call it, progesterone, corticosterone, testosterone, mainly when we get to the endocrine system. Don't worry about it for this test. They just modulate the activity of some of these central neurotransmitters, but we'll save that story for later. I'll just put it in here for completeness. You also get the idea that often all the neurotransmitters are either in the peripheral or central nervous system. There are many peptides that act as neurotransmitters and neuromodulators in other systems. And next semester, when we get to the GI system, you're going to hear about all kinds of peptides that control responsiveness of the GI system, such things as motility and secretion. And these agents are almost always small peptides. So we'll talk about glucagon and gastrin and cholecystokinin and many others that are essentially neurotransmitters in the gastrointestinal system. In fact, many of these were discovered before we even knew about neurotransmitters. We're going to talk about them and their function in the GI system. Again, don't worry about that now. One of the more interesting aspects of this whole story is some mood-increasing agents. We call them endogenous opiates. These are drugs that hook to the same receptors as opiates, like morphine and opium. These agents are produced by our cells, within our own cells, so we call them endogenous, and they have very natural analgesic, pain-reducing, and also euphoric, makes us happy, agents are producing neurotransmitters. In other words, they have mood-increasing activity and bind, like I said, to the same receptors. We wondered when we first we started using morphine and these drugs way before we knew anything how they worked, but once we found out that we had receptors for these agents, for morphine and so forth, these exogenous agents that increased our mood, we wondered why in the world we had evolved these receptors. Well, it turns out that we have neurotransmitters that hook to those receptors, and they're called endorphins and enkephalins, and they're increased with our own activity in interesting different ways. They're released in very strenuous activity like long distance running and swimming, and hence the addiction that some athletes have to continuous exercise and daily exercise. When they miss their exercise period, they get very depressed. And also excitement, tremendous excitement will release these agents, and there's been many cases where people have been injured in automobile accidents and are up helping other victims and helping them get out of automobiles and so forth, and don't realize till later that their arm's broken or they've got a fractured rib or something. They don't even realize it until they get out of the situation, and that's often because of the extremely high levels of endorphins in those activities. But we'll talk a little bit about them off and on. And finally, this last slide I just threw in here, these are the agents that I sometimes throw into a test question just to see what would happen if something was missing. So it might be useful for you to look at and think about what would happen with, I mentioned already, tetanus toxin or botulinum toxin. It blocks the release of vesicles. So what happens when these different agents are added to the system, and that's really how we've learned in most cases what goes on. Omega-conotoxin blocks the calcium channel, so you don't have the calcium channel coming in to cause vesicular release and so forth. The neuronal sodium channel is affected by tetrodotoxin. We mentioned that before. So anyway, you can look these over, and don't expect you to memorize them. If I ever ask you a question, I'll always say what that particular agent does. But it's a good way, and it's how we learned how most of these phenomena occur. Okay, let me start on the next lecture then. So this is a very general lecture that talks about the different parts of the nervous system before we get into more details. And also we talk about cerebrospinal fluid and its flow and things, so and different cell families within the nervous system. So it's a good place to start before we start going into details about the system. I already mentioned, I believe the nervous system is the acute communication system. They're differentiated from the endocrine system that also is our way to communicate with different tissues, but it's much slower and longer lasting. So this is like light switches versus temperature control or something like that in your home. The system is organized into motor and sensory and central activity, so we have all types of different sensations. It's what most of this unit will discuss, the different sensory apparatus, and then they conduct their information through sensory fibers to our central controller or integrative system and determines if we need a response, and then we have motor activity that goes up to the muscles and glands and generates responses. So we talk about sensory and motor. Sensory is often referred to as afferent, A, and E for motor, and that you can think of exit, if you will, because it exits the central nervous system. However you want to remember, efferent is motor activity and afferent is sensory activity. And visceral refers to, visceral refers to whether this system is going to internal structures or muscle and gland structures on the periphery, and we'll talk more about defining those later on. First of all, central nervous system is divided into a series of structures. The brain, the lowest part of the brain is the medulla, and that extends its most inferior aspect from the foramen magnum up towards the pons. So this is vegetative control area of the brain and controls all the contains all the tracts in the spinal cord up to the higher nervous system. Then the next level up is the pons and cerebellum. Pons contains tracts leaving the brainstem going up to the cerebellum and coming back from the cerebellum. And the cerebellum is the kind of the fine motor control of the system, as you'll see. Just above there is the midbrain that has mainly tracts going up and down, but also has some lower vegetative control systems. And then above that's a very large area that controls all kinds of vegetative activity called the diencephalon, which consists of the thalamus and hypothalamus primarily. And then the largest part of the brain, of course, are these two large cerebral hemispheres in the human, and they're divided into areas of different anatomical organization and function. And we'll save that for Unit 4 activity. And finally, the rest of the central nervous system is the spinal cord that goes from the and divided into areas that match the vertebral column, but it starts out from the foramen magnum, goes through a cervical, thoracic, lumbar, and sacral divisions of the spinal cord. The peripheral nervous system, in contrast to the central, can be looked at as also anatomically being divided into cranial and spinal portions. The cranial originate from and return to through the skull, through a foramen in the skull, where the spinal cords come through the intervertebral foramen, both outward for motor and inward for from sensory devices. And so we call this, it's coming out from the brain efferent and going into the brain afferent, cranial senses, and then spinal senses would be afferent, coming from the skin, from other sensory devices in the viscera, and efferent going out to the visceral tissues. We also can divide the peripheral system functionally. And so, again, there's sensory parts of the peripheral system. They consist of in some cases, very simple receptors, in some cases, very complex receptors or transducers that convert different types of sensation to a signal. And then they go through this sensory neuron or afferent neuron to the spinal cord. And then the motor activity comes from the spinal cord, and we talked about the primary motor neuron and how it couples with the neuromuscular junction. Well, this is somatic or an efferent neuron. And then there's another family of motor activity that's called autonomic. And the autonomic, as I just tried to explain in words a few minutes ago, consists of a first neuron. It's always a two-neuron pathway, which we call a preganglionic neuron, and then a second neuron, which we call a postganglionic neuron. And this is motor to the most of the visceral activities, smooth muscle and cardiac muscle and glands are innervated by autonomic nervous system. Now, there's a question on almost every test in physiology somewhere that asks, or in some way asks whether there's a sensory division of the autonomic nervous system. Although some authors disagree. Classically, the autonomic system is a motor system. There's an autonomic motor system, a somatic motor system, and then one large sensory system that provides information to both. Although people talk about visceral sensory receptors, classically they're in the same sensory pool as every other sensory. So the best answer is autonomic is purely motor, somatic is purely motor, and then there's one sensory system that provides sensory information from the whole to the whole central nervous system. I don't know why it's not going. Oh, there we go. Okay. I think this first phrase is getting outmoded. We used to say a neuron is the functional unit of the nervous system. We're going to talk about the glial cells in a minute, but they're just as functional as a neuron. But anyway, it's the primary cell, I guess we could say, of the nervous system. We've talked about it a lot, but now we're going to go into it a little bit more detail. Again, it consists of a cell body that surrounds the nucleus, so we call it a perikaryon or body. And then within that is a nucleus and nucleolus that's pictured here. The cytoplasm of a neuron contains neural fibrils, which are essentially cytoskeletal elements like in any other cell, but for some reason we call them neural fibrils, but they're mostly microtubules. And then the Nissl bodies, are named after the histologist discovered them by a stain, are essentially the ER of this cell. So it's essentially like any other cell, but it's unique in the type of integral proteins capable of generating action potentials and so forth, and it's an excitable cell. The dendrites are the short branching extensions from the cell body that extend the receptive surface. So you saw that figure a little while ago that showed all of the endings from all kinds of other neurons on this cell body and dendrites. So the receptive surface is this whole crown, if you will, of the soma. And it's responsible for receiving the neurotransmitter and converting it to a signal, and if it does so, it'll do it at the axon hillock. And so the axon is a single structure leaving the cell body. It conducts the impulse from the soma, And if you do a transection of an axon, you'll find it just filled with microtubules that almost looks like a root extending out with lots of tubules going through it. And it often has little side branches, which we call collaterals, that extends its network. And then the axon hillock, once again, is this cone-shaped device of the soma that extends into the axon. As I've said many times today, that's where the impulse begins, created by a neuron. We've also talked about myelin sheaths, but I want to extend it a little bit this time. Any cell in the nervous system that produces, well, all the cells that do are in the nervous system, any cell that produces layers of myelin, we call them neurolemocytes or neurolemocytes. And there's two families of them, Schwann cells and oligodendrocytes. Schwann cells are the ones that we've talked about before. And so Schwann cells wrap around peripheral neurons and form these segmented sheaths, as we before said, that allow a rapid form of neural transmission. And so this neurolemocyte is actually a Schwann cell that wraps around, puts many layers, and again, the places between each one of these are called nodes of Ranvier. Oligodendrocytes, in contrast, are only found in the central nervous system. I'll show you another figure in a minute, and their extensions do kind of the same thing, but I'll show you that in a better figure later. And again, this is the purpose of myelination, is to increase propagation velocity, and we showed you how that occurred. Neurons aren't all the same. They're definitely families of neurons. The ones that we're using all the time for models are actually multipolar neurons. They have many different dendrites and one large axon. And that's almost all the central nervous system neurons. But often the peripheral system neurons have different types of structure. Again, this shows nicely a collateral of an axon. A bipolar neuron looks like this. It has a central cell body and axon going one way and a single dendrite that might branch going the other way. These are found in all types of special sense organs. The one that is so clear and obvious is the olfactory tissue, the olfactory cortex, we sometimes call it, but that's our smell, our ability to smell. The sense organs for that look just like these and are bipolar neurons, but there's also bipolar neurons in the retina and other nervous structures. We'll point them out when we get there. There's a unipolar, sometimes called a pseudo-unipolar neuron. It's like the cell body stepped aside, and the axon and dendrite from the last one, a bipolar neuron, is now one solid structure. And these, again, are sensory neurons, afferent neurons, often in pathways from the receptor towards the central nervous system, and we'll point these out also when we get there. There are a tremendous number of other cells that are part of the nervous system, and although, like I said a minute ago, we talk about the neuron as being the primary functional cell, neurons couldn't function without all these different other cells. There's actually 10 times more neuroglial cells than there are neurons in the system. And we've learned a lot in the last 10, 20 years about these cells and what they do. And one of the most interesting of all of them is astrocytes. So Boren and Volpage spends a lot of time talking about astrocytes. I'm just going to try and summarize the story, but they're amazing cells. So this represents an astrocyte. Here it has a much smaller neuron, has all these processes coming out every side. Often it's very closely affiliated with the blood supply or capillaries. So one of the first ideas was they produce or are responsible for maintaining that blood-brain barrier in many cases, but they do a lot of things. This is a artist's kind of schematic. So this represents a capillary. Remember, the astrocytes are intimately around capillaries, and this represents a neuron. And a lot of the role of astrocytes is providing energy for neurons. You probably are familiar with the concept that without glucose, the brain shuts down in 5 minutes. In other words, there's no storage of glycogen in the central nervous system like there is in the body. We have a huge store of glycogen in the liver and in muscles, and so when we need more energy, we run out of glucose in the blood. The muscles and a lot of other tissues are well supplied by breaking down glycogen. Well, the brain has almost no glycogen, and all the glycogen has is in the astrocytes. So glucose, when it's present in the brain, which is almost always, goes right into neurons through a special transporter and takes care of the neuron's energy supply. But there's a second pathway in which glucose can go into astrocytes and be stored in glycogen and then be broken down into lactate and provide through lactate an anaerobic, if you will, supply of energy for neurons. This is the reason why we can survive even 5 minutes after running out of glucose. So brains can have a lactate supply for a brief period of time from this storage of glycogen. It can't last very long. That's why we go unconscious very quickly if glucose crashes. But there's now a fairly good body of evidence that shows that whenever our brain is very active and a lot is going on in our life, we always get a good amount of that energy from astrocytes. So this glucose supply, although probably going on all the time, is probably always appended or added to by this lactate energy supply from astrocytes. So this may be a really important way, a reason for astrocyte function to supply energy for neurons. Another very interesting thing we found out recently is astrocytes control a very important extracellular concentration of potassium. They have a very high, very polar membrane potential that's very sensitive to extracellular potassium. So the more polar cells are, the more sensitive they are to small changes in potassium. And they have the ability to store potassium when there's any little change in potassium. And you know how critical cell function is, especially excitable cells function, now it's affected by potassium. So it appears that whenever potassium goes up at all around neurons, that a lot of it goes into these astrocytes to keep the level low around neurons. And it's even better organized. Astrocytes are joined by gap junctions. And so if neurons very active, apparently the potassium concentration goes up around those neurons and lots of action potentials. This can be shuttled out of the area by astrocytes, taken from astrocyte to astrocyte, and released in other areas of the brain. So it's kind of buffers. They call it spatial buffering. It can move potassium around the cerebrospinal fluid such that it doesn't get high around any pool of neurons. They also produce and secrete some neurotransmitters. Not a lot's known about that. And so they're extremely functionally important, and we've just learned most of this in the last 10 years, and there's still a lot of study going on. I mentioned oligodendrocytes. So these are represented here by this A, I guess by the A, so that could also be astrocytes. I don't know which one of these. Oh, a C, it's my letter C, there we go. So oligodendrocytes, not a very good figure, that wrap around, much better here, they send their processes out and wrap around several axons in the vicinity. So rather than Schwann cells that wrap around cells, dendrocytes their processes wrap around several cells simultaneously. And again, they're only found in rows along nerve fibers and only in the central nervous system, where Schwann cells are the myelinating cell in the peripheral nervous system. Microglia, and here's a little, they call them microglia because they're the smallest of the family of glial cells and they don't have quite as many processes, are the macrophages. So it's just like I mentioned before, there seems to be a macrophage population for every tissue. And so these are phagocytic cells, and they show what happens when they get stimulated by an invasion. But anyway, they swell and engulf foreign material. I hope they're very active in people that are getting all these problems with meningitis going around these days. But anyway, that's their function to phagocytize foreign material in organisms. Ependymal cells, I'll talk about in just a minute. They form membranes within the ventricles of the brain and are responsible for forming cerebrospinal fluid. So they're like an epithelial membrane, essentially. They form a layer of cells, columnar cells, sometimes cuboidal cells, that on one side have blood and the other side secrete cerebrospinal fluid. So they're responsible for producing the cerebrospinal fluid, ependymal cells. And finally, another little comment about it's probably, I don't know why I put it here, but a comment about neurons. The protein synthesis primarily goes on, although there's a little bit of activity in the axonal ending. Most of it occurs in the ER, in the Golgi apparatus of the cell body, and then the neurotransmitter must travel out to the axon terminal to be released, and they depend on these axonal transport, these microtubules for that to occur. So they're put in vesicles out of the Golgi, and then the vesicles are taken to the axonal ending for storage and release, and empty vesicles are actually shuttled back for membrane sources back to the cell body. And this whole thing's called axonal transport. It's not only neurotransmitter, but also even mitochondria and other organelles are shuttled back and forth along this cable system, if you will. What is responsible for the activity is what we call molecular motors, that are small little proteins that move. Kinesins move out to the end of the ending, antigrade, and then the retrograde is dynin. They're two different, call them molecular motors, that move one direction or other, and they also hook to the cargo that's taken to these things. You'll hear more about this if you take cell biology as a whole section on molecular motors. And finally, the last thing we'll talk about today as far as this lecture is cerebrospinal fluid. It's kind of the bath that our brain sits in. So one of its functions is mechanical. A lot of times when you want to take care of something that's very soft and easy to fracture, you put it in a water bath. It's the same thing. The brain's sitting in this fluid bath so it doesn't get hurtled around too much when we jerk around in our little environment. It also has some immunological activity and chemical activity. Mostly here we're talking about the electrolyte content. It contains nutrients that help in maintaining the function of the cells and receives the byproducts of metabolism from these cells. And one unique thing about cerebrospinal fluid is it has a different constituency than plasma. We call it a blood-brain barrier. It has more lipids and highly lipophilic molecules, and also the interesting differences in, I'll get into in a second, into what molecules can cross and what can. I've already mentioned the choroid plexus, and here you can show it represented, this little red line that's part in the third ventricle. This membrane is in three of the ventricles, the lateral ventricle and third ventricle we call it, and produces cerebrospinal fluid, which begins as a filtrate of the plasma, but it's just not an ultrafiltrate, and that's brought in here. It has very low potassium, lower than even 2 to 3 millimolar, lower even than plasma, and very low amino acids, which is an interesting realization. Amino acids can easily cross most capillaries, but if we allow a lot of amino acids in the cerebrospinal fluid, many of them act as neurotransmitters, so it would disrupt the brain function. So we allow very little amino acids into the cerebrospinal fluid and essentially no protein. And the ion concentration stays remarkably stable, even more so than the plasma, not only because of this barrier, but also, again, because of astrocyte activity. There's probably think it's more than this, but there's only about 100 to 150 mils of cerebrospinal fluid. That's about a shot glass, I guess, about a small cup, if you will. And we reproduce it about 4 times a day. In other words, it's continually being secreted by the epidermal cells, then taken up by the venous system, as we'll show you in a second. So it's continually being resynthesized. So every 6 hours or so, we have a new cerebrospinal fluid circulating our tissue. To really understand the system, you have to look at the pathway that fluids can go in the brain. And so there are two, what we call lateral ventricles. And, you know, the ventricle heart is just a big compartment. Well, you're going to be dissecting a brain in a week or two, and you'll see the same type of things exist in the brain. There's these large holes, which we call ventricles. They're C-shaped, and there's one in each hemisphere. So these are called lateral ventricles, left and right ventricles in the hemispheres. And we also call them 1 and 2 because there's a third one that's right in between. It's called the third ventricle. So we don't really decide or care which one is 1 or 2. But they empty into a third ventricle via interventricular foramen. So right here in the arrow, or right here you can see them, is what we call the interventricular foramen. That is how the fluid gets from the lateral ventricles into the third ventricle. And the third ventricle lies right between the two hemispheres in an area below what we call the corpus callosum that we'll show you in a week or so. And then it is connected to a fourth ventricle by a little structure called the cerebral aqueduct, which is shown right here. So here's the third ventricle, here's a fourth ventricle, which is right inferior to the cerebellum, and the third and fourth are connected by the cerebral aqueduct. And then the fourth ventricle is the way that the cerebral spinal fluid either goes into the central canal of the spinal cord or around the brain through what we call a foramen of Luschka. That I'll show you in a better figure. So here's another way that you can imagine the whole thing. To orient you, this is a sagittal section. So here's the two lateral ventricles. They empty into the third ventricle right here via the interventricular septum, or foramen, which is also called the foramen Monroe, just to confuse everything. So here's the third ventricle. It opens into the fourth ventricle through the aqueduct of Sylvius, or everything has to have two names, cerebral aqueduct. And then this is the fourth ventricle. Fluid can exit the fourth ventricle two different ways. It can go into the spinal cord central canal right here and down through the spinal cord, or it can go through these two foramen, which we call the foramen of Luschka, that go around the brain in what we call a subarachnoid space, which we're going to also show you in another figure. And coming into the subarachnoid space are some little venous villi, which we call arachnoid villi, that absorb spinal fluid, take it back into the plasma, and then later it's resynthesized and comes out that those structures, as we said before. So anyway, that's kind of a pattern of secretion of the structure. And here's another picture just to give you a couple of figures that show the same thing in the cerebral spinal fluid. This is a very confusing diagram, but it makes the point that a lot of students never get, that there is a blood supply to the brain. Sometimes you think that the cerebral spinal fluid is supplying all the nutrients and all of the removing the waste products, but there is also a circulation. There are blood vessels going through the central nervous system. And so this is the production to the ependymal cells of the cerebrospinal fluid, and then absorption to the arachnoid villi into the venous system. So this is what we just talked about, the cerebrospinal fluid circulation. But there's also arteries that that come into the brain and then produce some of the extracellular fluid that surrounds the cells, and then you have the same venules and veins and so forth that you do in any other blood supply. It's just that these are very tight capillaries and don't have a very lot of filtration capacity like other capillary beds do. So the intracellular compartment of neurons and neuroglia is kind of a component of both the fluid supplied by the blood vessels as well as the cerebrospinal fluid. So these are two parallel supplies. So this one, a lot of students never even get the point of, but there are definitely blood vessels going to the same area that the cerebral spinal fluid is present in. Okay. So, last thing before we go to our review, if you want to come today, is some iClicker questions. So, just to keep us up to date, a couple of iClicker questions. First one of my favorite long questions. So, muconotoxin is on that figure that I showed you, all those different toxins, that blocks voltage-gated sodium channels, but only in skeletal muscle. It doesn't affect neural action potentials, but seems to only affect skeletal muscle action potential. So if you administered to an animal an effective dose of muconotoxin in an experiment, which step would be blocked? So here we have a motor neuron coming around, and we can measure EPSPs and so forth. Which step would be blocked? It's a pretty obvious question. Don't think about it too much. Okay, I think I had 45 seconds, so there's 10 seconds left. 7, 6. Okay. Let's see what she chose. Pretty scattered. It was just meant to be a really easy question. The only thing blocked is the muscle action potential. So the nervous action potential is normal. You have release of the neurotransmitter. You have an excitatory post potential, end plate potential, because that's mediated by the acetylcholine receptor. So the only thing that doesn't occur is a muscle action potential. So D is the correct answer. Number 2. The figure below represents an axon hillock recording from a multipolar neuron. The blue, red, and green lines represent increasing number of presynaptic endings. So each of these, this is 4 endings, this is 8, this is 16. What type of neuronal integration is this illustrating? Okay, 6, 5, 4, 3, 2, 1. It's like almost everybody got the point. And 89% did it. Spatial summation, this is simply adding more and more neuron or presynaptic endings, so that causes the axon hillock to fire. So that's classic spatial summation, so that's letter B. Most of you got that. Number 3. Which of the following increased the excitability of a neuromuscular junction? So it would cause that neuromuscular junction to be more excited than normal. Okay, And that's the time. Okay, so tetanus toxin, I mentioned before, blocks the vesicular release, so that certainly wouldn't make it more excitable. Omega conotoxin blocks those presynaptic calcium channels, so calcium couldn't get in and release the neurotransmitter, so that's not right. Saxitoxin blocks voltage-gated calcium channels, sodium channels, and so the sarcolemma, the muscle membrane, can't have action potentials. Tubocurare is also an acetylcholine receptor antagonist. So nicotine actually activates the receptor. An agonist, once again, activates the receptor. Everybody got that, so almost 95%. Okay, so D is the right answer. Number 4, talked about these last time. Miniature end plate potentials, MEPPs. This on an exam a couple of years ago, and hardly anybody got it right. I thought it was easy. Okay, 7 seconds. 5. And pretty good. I thought you guys would do better. EPPs that are recorded on the motor end plate, just a little blitz, because we continually lose vesicles all the time from the presynaptic endings. So D is the right answer. Spontaneous neurotransmitter vesicular release occurs constantly, so there's always a little tiny bit of end plate activity. So D is the right answer. And finally, number 5. Electrical impulses or gap junctions, where are they found? I've talked about this off and on. One of these places doesn't have gap junctions. It's a Sesame Street question. Which of these is not like the other? Okay, 10 seconds. 5. Okay. And the right answer is, wow. Maybe you read it as present or not absent. There's definitely gap junctions in heart cells for sure, and any smooth muscle tissue, in glial cells. I just talked about them in astrocytes, and I talked about them in bone. So definitely not neuromuscular junctions. Anyway, no wonder all my students missed it. Okay, so anybody that wants to look at your test, it's in that bracket. I'll see you in room 124.